I'm going to introduce Dr. Paul Nelson here for you guys. An incredible man. We really appreciate the fact that you're here. Thank you so much for coming. I know Pastor Daryl Lindensmith, who actually is in charge of this, is super thankful that you're here, and we are too. So thank you so much for coming. Dr. Paul Nelson is currently, a fellow of the Discovery Institute and adjunct professor in the Master of Arts program in Science and Religion at Biola University. He's a philosopher of biology who has been involved in an intelligent design debate internationally for over 2 decades. After Dr. Paul received his BA in philosophy with a minor in evolutionary biology from the University of Pittsburgh, he entered the University of Chicago, where he received his PhD in 1998, correct? In the philosophy of biology and evolutionary theory. We're super thankful to have you here, Dr. Paul Nelson. Thank you so much again. We welcome you up on stage. Thank you. Yeah, give him a round of applause. And if we can, if you don't mind, we'd actually love to start out with prayer, just to ask the Holy Spirit to be here with us tonight. All right, Father in heaven, we ask for your presence. Ultimately, we're here to see if who you say you are is actually real. So come out, show yourself to us. Don't hide yourself from us. We know that you'll be here, so we ask for you to show up and to speak. In your name I pray. Amen.
Well, I'm delighted to see all of you. How's this? Is this a decent volume? You can hear? All right. And I know this title probably seems a little cryptic to you, but by the time I'm done, it should all make sense. So, what are orphan genes? What's the tree of life? The tree of life is Darwin's picture of the history of life on this planet, in which every living thing is related to every other living thing. So I have some of my relatives here who came from Horace to see me. My mom grew up in this area. But all human beings are related. According to the tree of life, you're related not just to every other human being, but in fact to your dog and to the trees and to the gut, to the bacteria in your gut, right? So every living thing is connected in this great tree. Now, this theory is widely accepted by modern biology, but within the past decade and a half, it's come under increasing scrutiny, skeptical scrutiny, because of discoveries made in genetics that no one expected. Certainly, I didn't expect them as a graduate student studying evolutionary theory. And these discoveries are incredibly interesting and significant. So I'm going to describe them to you tonight. And if you want to contact me later, I can give you more information, probably more than you would ever want to read about the subject. But let's start with this question. If this theory, this tree of life theory, weren't true, how would we know? How would we know it? Now, that's an important question to ask, because you don't want a scientific theory to be impervious to testing. Even a widely accepted scientific theory might end up being false, and if it is false, we would want to know, how could we discover that? Well, that question has become increasingly important, as I said a moment ago, within biology. But before we get there, just to kind of warm you up, it's a cold night here in Fargo, let me give you a metaphor for thinking about these kinds of questions. So we're here in Fargo, and let's say I meet you at a Starbucks, right? We're sitting there having our coffee and chatting, and I said, you know, I walked from Fargo to Grandin. All right, that's about 28 miles. Grandin's due north of us, and you think about what I'm saying, and you say, I could believe that. You know, 3.5 miles an hour, normal walking speed for an adult male, otherwise healthy adult male. You could do that in about 8 hours. So you have no problem believing my claim. You have no reason to doubt it. All right, same Starbucks a week later, or let's say a couple weeks later, I see you, you've got your coffee, I sit down, and you say, you know what, I walked to Dickinson. Now you're a little more skeptical. That's 280 miles. And you say, well, you could have done that, but there's no way you did it in a single day, like you said with Grandin, and you certainly needed help. Meaning you needed food, you needed water, you needed shelter, especially at this time of year you would need shelter. You could do it, but never in a single day. It would take probably about 10 days, maybe a little longer. Yeah, it's possible, but I have reasons to be skeptical. All right. Same Starbucks, same scenario, and I tell you I did that. All right, now you have no reason to believe me. In fact, you may be looking at me with, considerable worry, like this guy might need some psychiatric attention. Because when I hit the Pacific coast, I run into that, all right? And in its normal state, you're not going to walk on it, right? You are not going to get from the coast of California or Oregon to Honolulu. It's just not going to happen. You need a boat or an airplane or some other means of transportation other than your two legs. All right, so how do we evaluate claims like this? Well, we know what walking takes. For an adult human being, we know what walking requires. You have to have a firm surface. You've got to be in reasonably good health and so forth. You're going to go any distance. You're going to need, some support. And we have maps of the terrain. We know that when you get to the coast, the Pacific coast of the United States, that there are several 100 kilometers of water between you and Honolulu. All right, so we know what the terrain is. Having that background knowledge lets us evaluate the plausibility of claims about what people say, when they claim to have walked somewhere. Well, the same is true for biology. Okay, we evaluate claims in biology, particularly in evolutionary biology, with respect to what do organisms need just to exist at all. What do they require for viability and normal function? And if your theory says, well, in this particular evolutionary transition, you need to be dead for 5 minutes, on those grounds alone, we can rule out that theory. No, it's not going to work. You've got to be alive to evolve. But we also have maps. We understand where the terrain is. And this is where the discoveries that I'm going to describe in a few minutes become so significant, because they're showing us that the genetic diversity of life on this planet is vastly greater than anyone realized. Vastly greater than anyone realized. To get you there, I want to take you through a familiar sort of line of argument or thinking that confronts all of us all the time, and that is, why aren't we chimps? Okay? So here is hemoglobin, delta hemoglobin. This protein is circulating in your blood, carrying oxygen, and the very same protein is circulating in the blood of chimpanzees. Okay, so on the top, we see the human sequence, amino acid by amino acid. These are single letters representing individual amino acids. So you can think of this like a very long sentence in protein space, if you will. That's the sequence for human delta hemoglobin. Here's the sequence for chimp delta hemoglobin, and they're nearly identical. There's one difference. So at that position, we have the amino acid methionine, and they have, I think it's a valine there. All right? Otherwise, identical. But there are lots of proteins in us and in chimps that are exactly identical. So if I gave you the proteins and I didn't tell you what species they had come from, you wouldn't be able to tell me on the basis of those proteins whether they came from a chimp or from a human, which raises an interesting question. If at that level of proteins we're so close to chimps, why don't we look like this? And I want no smart remarks about any of your friends who, may resemble those creatures. There are no chimps here tonight to hear this talk, okay? When I go to the zoo, it's always the chimps in the enclosure and the humans walking around looking at them and so forth. We don't look like this. If you're lucky enough, you look like that, right? A couple of actors in a movie a few years ago. There are lots of differences between us and chimps in terms of our overall biology, and they're remarkable when you start compiling them, okay? So we don't have a sagittal crest. If you run your hand over the top of your head, you don't feel a little ridge there. Chimps do. We have chins. Chimps don't. You can always tell when you're watching a science fiction movie, as if you didn't know anyway, like Planet of the Apes or 2001: A Space Odyssey, that it's a human being in a chimp suit, because the sclera, the white of the eye, will be white. With all the other primates, chimps included, their sclera are colored. All right? Significant difference, minor, but very detectable, very distinctive of human beings, and on and on. For the men in the room, chimps have a bone in their penis. All right, the men in the room know that's not true for us, right? There's no bone down there. So we could just, pile up all the differences, and these, this could go for many pages. These differences at the biological level, at the level of organisms between us and chimps. Of course, when you consider behavior, there are all kinds of differences. Really, the most striking differences in terms of things such as getting sick. When a human being falls ill or becomes elderly, normal human population, we care for our elderly, we care for our sick. If you're in the chimp population, you're out of luck. They'll just leave you behind. We store food in a way that no other primate does. We cultivate food in a way that no other primate does, and so forth. So we've got our puzzle. If we're so similar genetically, why are we so different biologically? And this has been a puzzle in evolutionary theory for a long time. For at least 40 years, probably longer. In fact, I remember as a student reading this paper in the journal Science. Very influential paper, published in 1975, by two scientists at Berkeley, and they said if you look at any protein in a chimp, it's largely identical to that in a human being, yet when you look at the organisms as a whole, look at their behavior, look at their anatomy, and so forth, they're remarkably different. In this paper, they say any bone in a chimp can be distinguished readily from any corresponding bone in a human. So there's a puzzle for evolution. What's going on? Why are we so different? Jonathan Marks at the University of North Carolina said, well, you know what? The genetics isn't telling you anything interesting. He said, if the overall biology of the organisms tells you that they're very different and the genetics tells you they're largely the same, the genetics is not telling you what's important in what makes a chimp and a human. So I'm paraphrasing what he says here. And in more recent publications, he's been even more outspoken about this. He says, I have no trouble recognizing the humans. The humans are the ones building buildings. They're the ones praying. They're the ones going to the moon. Chimps aren't doing any of that. That's kind of a cynical view, but I want to take a different view that applies to our topic tonight. Evolution, as a scientific theory, is not mainly a theory of similarity. Okay, similarity just gives you more of the same thing. Evolution fundamentally is a theory of transformation. It tells you how to go from A to B, how to transform from a common ancestor, a lineage, one branch of which leads to chimpanzees, another branch of which leads to Homo sapiens. If you don't have something that explains this, that explains that process of transformation, you don't have a theory of evolution. That's what evolution fundamentally claims to be able to explain. And Darwin understood this. In his most important piece of writing, The Origin of Species, he never actually uses the word evolution except once on the last page of the book. He actually uses the verb form of it. His own preferred term for his theory was this, descent with modification. And even though it's longer, in many respects, it's a much better description because you've got two elements here. That gives you similarity, right? So I have my cousins here. They resemble their parents, and their parents resemble their parents, and so forth. There are these family resemblances that we have in lineages that give rise to similarity. So that's where you get your similarity from, but similarity alone is not evolution, as I said a moment ago. This gives you evolution, that aspect of transformation, of changing one species into another kind of species. And Darwin understood that this mattered, because if he couldn't explain transformation, he couldn't have modification, and he really couldn't have evolution. In fact, in The Origin, he proposes a test. He says, if you could show me any characteristic of a living thing that couldn't be explained by a pathway of transformation, that he calls numerous successive slight modifications, he says my theory would absolutely break down. So he's aware, I've got to be able to show you how to change species, how to transform them, and if I can't, my theory won't work. So he's well aware of that. That is not evolution. All right, now, let's start to approach this topic of orphan genes and why they're so significant. What I'm going to describe is work that I did with two people in particular This guy, Lee Van Valen, who died in 2010, served on my PhD committee, and then he and I, for several years after that, talked about this topic on a regular basis. I was working on a major piece of writing with him, and unfortunately he fell ill and died, but I want to acknowledge his influence in my thinking. This scientist, who's a botanist in the United Kingdom, is very much alive, working in London. And I looked at two questions with both of these guys that ended up being closely linked to each other. The question I looked at with Lee Van Valen is this: if common descent were false, how would we know it? Now, that's the question I've already introduced. With Richard, I looked at the question: what is the role of orphan genes in biology and evolution? Now, this ends up being very significant to that. So these questions are closely connected in an interesting way that I hope to show you in just a moment. But before we get there, let's talk about common descent in a little more detail. Here's a textbook I used as a student. Okay, undergraduate studying evolutionary biology. Look at the page number here, page 1. You are entering the field of evolutionary biology through the gateway of this claim that all living things are related. So in a sense, it's the fundamental assertion of the theory. It's the first thing you learn, that not only are you related to every other human being, but you're related to your dog and the trees and the bacteria in your large intestine. And typically it's represented this way. Now this does not look like a tree, I'll give you that. But imagine that you're in a helicopter hovering over a very large oak tree. Okay, so you're looking at a tree top down. This could be a very large oak tree here somewhere in Fargo. And here would be the root where it goes into the ground. And you've got 3 major branches spreading out from that root, 3 major sections to the tree. We're in this one right here. We're on the little branch here marked animals. So everything you see on Animal Planet or the Discovery Channel or on a National Geographic program, all the warm, fuzzy things and the slimy, scary things, they're all on that animal branch, which is really only a tiny portion of the full diversity of life on this planet. These 2 big branches are single-celled. These are organisms that do not have nuclei. They don't have a nucleus in their cells. All the organisms here, as their name actually means, have a nucleus. Okay? The fundamental claim being made here, though, is everything on the planet falls somewhere on that tree, and there's nothing outside it. Now, that's a strong claim to make. You could think about biology as a science of exceptions. Okay, so I tell you, hemoglobin is the oxygen carrier for organisms, except when it's not. There are some organisms that have hemocyanin, which is a different protein. So for almost any claim that you make in biology, you can find an exception that breaks that rule. With this theory, it's truly universal. Okay, that black line, that circle indicates if you're a living thing alive today or at some point in the history of the Earth, you're going to fall somewhere within that black circle, somewhere within that domain, and there's nothing outside it. Now, for scientists, that kind of universality is very important because it allows you to compress a lot of experience down into a form that you can stick in your pocket and take out into the world with you. And I want to acknowledge the power of that for many biologists. They like, and scientists in general, they like theories that have that kind of power. But your ultimate grandparent on this view is here. It's the last universal common ancestor, often abbreviated LUCA. If this theory were true and you could trace back your ancestry 3 billion years, you would find LUCA present in the set of all of your ancestors, and it's the organism present in the set of everybody's ancestors. In fact, it's the only one. And until quite recently, as I said at the beginning, this theory was viewed as just something you didn't doubt, right? If you were properly educated at all, not just in science, but properly educated at all, this was part of the furniture of reality. There was really no reason to doubt it because it looked like all the evidence supported it. So in the mid-'80s, Francisco Ayala, who's a geneticist, University of California, he said it's like the roundness of the Earth, right? It's just a fact about reality that we have no reason to doubt. Now look at the strength of that claim and compare that to this, okay? in January 2009, New Scientist magazine, it's a science publication published in England, had a cover story, Darwin was wrong, cutting down the tree of life. Okay? If you had told me as a graduate student that a publication like New Scientist would bring out this kind of article, I never would have believed it. Because when I was a student, I was taught that claim of Ayala's, namely, you don't doubt this theory if you're properly educated. And here they're saying, just a few years later, it's actually wrong. Now, the writer who wrote that article was attacked on the web by leading evolutionary biologists. They said, we're never going to have anything to do with your magazine again. You're just catering to creationists by publishing a story like that. And he defended himself and he said, no, I'm not. I don't care about the creation evolution debate. I'm simply describing what's going on within evolutionary biology. For instance, looking at the work of this man who died a couple of years ago, very influential scientist who actually gave us that 3-domain tree picture of the history of life. He was at the University of Illinois, Carl Lowe's. I met him when I was a student. And in a publication that you can actually download off the web, it's open access now, he says biology has to go beyond what he calls the doctrine of common descent. In other words, the single tree picture just isn't true. And that capital D there in doctrine, that's not a typo, that's his term. And for Lowe's, a doctrine of that sort is something standing in the way of scientific understanding. All right, so there's a revolution ongoing right now in evolutionary biology, casting doubt on Darwin's single tree picture. All right, but let's go back to Darwin's test, because it's that's going to show us why these orphan genes are so significant. All right, Darwin said, you know what? I think everything fits within this single domain, right? All living things are related. And by the way, this cartoon here, this picture, you could never publish something like that. That's just a diagram to give you this image of a singularity at the root of the tree of life. But that's not a proper evolutionary branching tree. It's just a PowerPoint cartoon. All right, so Darwin says, show me any feature of a living thing that could not have arisen along a natural pathway, and my theory would absolutely break down. So let's take just one of those pathways. Let's take this one right here. Okay, A goes to B. Now B evolved, according to the theory, along this pathway, starting at A, the last universal common ancestor, but it's still within the domain. It might not be. You could have a point outside the domain that if you saw it and there was no natural pathway connecting it to the rest of life, you would have reason to doubt that this was true, that this picture was true, because you've observed, let's call it B prime. There it is. That natural pathway doesn't exist. And you say, well, you know, it's out there. It's not connected to the rest of life. I have now good grounds to doubt that this theory is really universal, because that's, after all, its major claim, that every living thing is related. So that would falsify common descent. Now, this has come to have a name within evolutionary theory. It's called the principle of continuity. And once you see it, as I did as a graduate student, and become aware of its role in the thinking of biologists, you see it absolutely everywhere. Because it's a key tool that evolutionary biologists use to judge the plausibility of each other's theories. And it's simplicity itself. Okay? So suppose I say to you, B evolved from A along that pathway indicated by the arrow there. I make that claim. That's how B came to exist. Well, you're not, remember my Starbucks story. Maybe you'll believe me, that's not a very great distance. And then I say, well, B is a brand new feature, it didn't exist before. Now you're a little skeptical, and you're going to bring to bear everything else that you know to evaluate my claim, just the way you did with me claiming to walk to Honolulu. You say, well, you know what, Paul? I can see points along this pathway that are not biologically possible. That could not have happened. That claim that you're making violates the principle of continuity. You've got to be alive all along that path. I can see a point on that pathway where you're going to have real functional problems, so I have reason to doubt that this actually happened. That's how the principle of continuity works, and it's tremendously powerful. And it's used everywhere in evolutionary biology, sometimes by that name, sometimes just implicitly. But there's a logical problem. If I have a universal theory, how could there be anything outside it? I've formulated it in such a way that everything already falls within that dark black circle, right? Every living thing, T-Rex, oak trees, blue whales, starfish, they're all within there. What could be outside? Now, Darwin himself and his intellectual offspring, evolutionary biologists in the 20th century, solved the problem by using a kind of proposition called a counterfactual conditional. Now, don't let the name intimidate you. You do this all the time. If you have teenage children, you use it all the time. If you do this, this will happen. Okay? You probably don't give it that name, counterfactual conditional, but you know the logical form. This is what you propose to do. It hasn't happened yet, but if you do it, this will happen, okay? So what we're going to do is we're going to fill in X here. So biologists say, well, yeah, we've already dumped everything into the domain of common descent, but I can imagine observations that if I saw them would give me reason to doubt the theory. So that's what they do. They fill in X there with some hypothetical observation, which if they saw it would give them reason to doubt the theory. So they say, for instance, we should not observe a lot of unique genes and proteins that are unconnected to the rest of life. Okay, these are the orphans. We're going to come back to this, all right? But here are some other counterfactual conditionals that were made. I remember reading them as a student. For instance, down here, when I was a student, undergrad, 1980 to 1984 at the University of Pittsburgh, my textbook said there should be only one genetic code. The genetic code is the translation table that takes us from the realm of DNA and RNA to the realm of protein. And it's a true code, right? It's not chemically demanded. It's actually a true code. And at the time, in the early 80s, it was thought there was only one code for every living thing on this planet. We all used the same one because we all inherited them from the last universal common ancestor. Now there are so many different codes that you have to go look them up. If you're using a particular organism that has its own special code, you will make mistakes in biotechnology if you don't use that special code for that organism, and instead you use the so-called universal code. There's a web page where you can go and you can look up all the different codes that now have been discovered for life. And you know what happened? This domain swelled like a balloon to take that in. I won't go in, for the sake of time, I won't go into these other examples, but that's not a healthy position for a science to be in. If you make a prediction and the prediction fails, that means something. And if you let your theory expand like a blob to take in that failed prediction, you will rob your theory of its content. All right, so in the case of this, though, unique genes and proteins, the blob didn't get any bigger. And biologists started to say, I don't buy it. I don't think there ever was a single tree of life. And we'll come to those guys in a few minutes. Now, you might think, I've got this domain, every living thing's in there. I've probably done a good job of searching that space. That's probably been pretty well mapped out. In fact, only a tiny portion has been searched. And we realized this, biology realized this in the mid-90s when this technology came on stream. When I was a student, for most of my graduate studies, automated DNA sequencing didn't exist. When it began to be a rapid and relatively inexpensive technology so that you could sequence all the DNA of a species, we discovered things that no one expected. And I'll tell you something, this technology is becoming so inexpensive that high school biology classes in the next decade will have their own DNA sequencers. And for a semester project, they will sequence the DNA of a species nobody else has looked at. I have an acquaintance in the United Kingdom who's working on a kind of technology that's about the size of a TV remote. The whole device fits in your hand, and this is actually a much older machine. So this is not going to change. This is something that if you had told me again as a graduate student, I never would have believed it. But here's what's happened. Our knowledge of the genetic diversity of life on this planet has exploded. Okay, so I'm a student, 1980 to 1984 at Pitt, then I go to Chicago. I defend in 1997. Most of what we know about the genetics of life on this planet has been learned since then. This huge blue spike. And this figure only goes to 2008. I need to update it because actually this curve now goes up through the ceiling. Meaning, we really didn't know what was out there until we got this technology that let us look. And it's that enabled the puzzle of orphan genes. So, what's an orphan? Well, you know what an orphan is in English, right? It's a kid with no parents. There's a pun in the name, though. An ORF in molecular biology is an open reading frame. ORF stands for open reading frame, and it's a sequence of DNA that codes for protein. Now, the ORF that I've shown here, this is a little cartoon, this is what you would find in a bacterial cell. It's very well defined. It has a site, this is a sequence of DNA where the DNA tells the RNA polymerase first, and then later the ribosome, start here, go through this sequence. This is going to form a protein. And then you come to a site that's a stop. It tells the machinery, stop. All right. And these features are well enough defined that you could write algorithms and get computers to go looking for them, which is what scientists who work in bioinformatics do. So think of that like a long word, a long genetic word. And the parallels to English actually are very strong. We'll look at some of them in just a moment. So here's my new sequence of DNA that I've just determined from some species I've never looked at before, and I submit it to GenBank. Now, GenBank is the computer database that stores known DNA sequences. It's paid for by your tax dollars. And GenBank has open web pages. You can go and browse through there if you have nothing else to do. And if you do that, you truly are a nerd. Okay, so this portion of the cartoon, this represents sequences that are already in, let's call it the genetic dictionary. And I've got my new sequence, and I go looking for a match. Just the way that if I gave you an English word you'd never seen before, you'd get out your Funkin' Wagnalls or your Random House Dictionary and look for the corresponding character string to get the meaning. So in this case, we get a match. There's a high degree of similarity between the sequence that we just determined and the one that's already stored in the genetic dictionary in GenBank. And that's going to tell us now what this gene is doing, because that one codes for a protein that goes in the machine that makes proteins, the ribosome. Well, that tells us what this one is doing. And now we can add this one to the dictionary, and the dictionary gets bigger and bigger and bigger and bigger and more adequate, except with orphans, it doesn't. With orphans, there are no matches. You've got your DNA, you put it into the software that does this matching for you, and you get a report back that says no significant similarity found. It looks like a gene. We don't know what it's doing or why it's there. It's a puzzle. So when this was first noticed, and it didn't take very long for this to pop up as a puzzle, commentators like Russell Doolittle, the University of California, said, where's all this coming from? We did not expect so many genes to be unidentified. We thought we'd be able to do it just by computers alone, right? Get the new DNA, feed it into the matching algorithms, look for the matches, build our genetic dictionary that way. He said, we've got all kinds of mysteries here. Where are these genes coming from? All right, so he's looking at the signal right about there, right? Now it's very much stronger, but it's the same puzzle. He goes on, he says, we didn't expect this because we thought we understood where new genes and proteins came from. What's going on, he said, these are baffling mysteries. We don't really know where all these genes are coming from. I'm going to skip this one for the slide for sake of time. But orphan genes are ubiquitous. So let's say we've got a species and we've sequenced the DNA, and some of it gives us matches. We put it into GenBank, we get the matches back, that's clear. But then there's a fraction that doesn't, and those are the orphans. And they are a significant fraction of the genome of every species on this planet that's been looked at so far. So a year after Doolittle makes his commentary, this Israeli team, also working on the puzzle, look at it and they say, what's going on here? If evolution works through descent with modification, in other words, you get new genes and proteins from old genes and proteins by well understood evolutionary processes, where are all these orphan genes coming from? This woman, for a while, maintained a web page that she called the Orphanage, where she was trying to keep track of all of these unique genes and proteins, and she stopped because there are just too many of them. And Doolittle, then 2 years later, he said, we thought this could be done in more or less an automated way using computers because we expected, look at the tree of life. If you've got a protein and a gene in your species, it can trace its ancestry back to the set present in the last universal common ancestry. Along some very long evolutionary lineage, every gene and protein in you has parents. They're not orphans. And we should be able to build that family tree of genes and proteins, working our way back through life. So this is what's in the back of his mind, right? Here's a gene sequence that codes for a protein that looks like that. This sequence is subject to various evolutionary processes. It undergoes some changes. The protein it codes for then changes. Maybe you add an alpha helix right there. But there's still going to be a lot of similarity between these two genes. And these proteins have the same backbone, except this one has an added structure there. So this is what he's got in the back of his mind. You should be able to do this for any gene or protein on the planet, if this picture is true. Okay, so here we are, Homo sapiens. That's you and me, and we look at our genes and proteins, and we start working our way back down the family history of our biology, back down to the minimal set that was present in Luca. So that's what he has in the back of his mind. That common descent picture is true. It's not up for grabs. We think we know how genes and proteins arise. If 1 and 2 are true, the very last thing you would expect is a lot of unique genes and proteins, which is, in fact, in 2016, what we observe today. And then in a few minutes, I'll come to some examples that will blow your mind. But Doolittle's not the only person noticing this problem. Kenneth Weiss at Penn State, in a recent publication, said, where are these orphans coming from? They don't fit our picture of the history of life. So there's an evolutionary puzzle, but it has all kinds of dimensions. Let me give you just a couple. One of the things you can do when you've determined all the DNA of a species, so here's a circular chromosome like you might find in a bacterial cell. Most prokaryotes, like bacteria, have circular chromosomes. Some have linear. We have linear chromosomes. But if you've got that whole sequence, using a piece of DNA called a transposon that randomly integrates itself into other DNA, you can knock out those genes to see what will happen. So we put the transposon in those cells, and here these red hatch marks show the places where it's integrated itself, and those cells die. Because at least one of those transposon hits knocked out an essential gene that cell couldn't do without. All right, so you won't see anything on your plates when you put these bacteria on. There's no growth. They're dead. Same thing here. At least one of these hit an essential gene. Sometimes, however, the transposons will integrate and the cells will grow. You'll have activity on your plates. You'll see colonies of bacteria. The very fact that the transposon is there in DNA in a living cell tells you when it integrated there, it didn't hit anything essential. So whatever that location is, those genes probably you can do without, at least under laboratory conditions. Well, if you do this often enough with these colonies that are alive and you work your way around that chromosome, you figure out where the essential hardware is. Now, this experiment has been done several times with different species, and the results are always amazing. So I'll talk about just one of them. This was done at Stanford in Lucy Shapiro's lab using a kind of bacteria called Colobacter that you can culture out of fresh water. So there's Colobacter in the streams and rivers here in North Dakota. They did the experiment and they found that in the essential hardware for that species were 90 segments of unknown function. And Lucy Shapiro herself, she's the senior author on the paper, she said, this is amazing. We found 90 genetic components. We have no idea what they're doing, but there they are in the essential hardware for this particular species. Now, why is that significant for evolution? Well, here's why. This is a figure from their paper. This is the bacteria in your large intestine. You have trillions of these cells in there right now. They're essential for the normal digestion of a human being, E. coli. very well studied. Colobacter is supposed to be a relative of E. coli in an evolutionary tree. They're related. They're in the same bacterial phylum. Now look, Colobacter has a lot of hardware that is essential but belongs just to it. E. coli has an even larger fraction of essential hardware that belongs just to E. coli. What that means is these cells are not running their biochemistry the same way. Just as if we went to an auto show here and we had a whole bunch of, let's say, Mustangs, Ford Mustangs lined up, we popped the hoods and we worked our way down that line of Mustangs, and every engine was different. Yeah, some parts would be the same, but every engine had essential parts that belonged just to that engine. The thing you would naturally infer from a situation like that is there's been significant re-engineering or tinkering with these engines because they're not operating using the same hardware. That's what we see in this picture. This species and this species of bacteria, even though they're supposed to be related, are actually running their biochemistry using different bits and pieces, which is not the story you would get from a molecular biology textbook, at least not one published in the past decade. All right, before I get to this example, I'm going to show you how old I am. Who at home has a telephone with a spiraling cord that connects the handset to the base? Please, someone raise their hand, okay? Be honest, okay? Marty and I are the same age, so I know. All right, there's a photograph of the top of my desk in my study in Chicago. There's my office phone. Okay, this is what my office desktop normally looks like. What's happening here? You know what happens. If you don't periodically disconnect the handset from the base and let out the supercoiling in the cord, you get a big knot. And with this phone, which drives me crazy, if I don't do it twice a week, I can't pull the handset up to my ear when the phone rings. Because of the geometry of the cord, you get this supercoiling. There's a nice one, right? That's what you don't want to see in your phone cord. Okay, DNA, because of its geometry, has the same problem. It's a spiraling double helix. And if you don't have proteins in your cells that can disentangle your DNA, you will die. They're called topoisomerases. Their job at the cellular level is to disentangle DNA. There are two different major families of topoisomerases. Topo1s make a cut in one of these backbones. Okay, Topo2s cut both of them. But what they do is they let out the supercoiling so that the cellular machinery that processes DNA can get at it. Keep in mind they're essential. Every cell that we know on this planet has them, and if you don't, you'll die. All right, now, this species has its own topoisomerase. As far as we know, this unique protein is found in a single species, and it's a species that lives at very high temperature that would cook you or me, right? You would not, it's just not like a nice whirlpool. This is really hot, okay? But it thrives, and in fact, this topoisomerase, because it's stable at high temperature, is useful in a lab kit where DNA is processed at high temperature. But when this was discovered, the French scientist who was commenting on it said, this is mysterious. Where did this come from? Where did this protein come from? It's found in a single species. In fact, in the article, he says, how mean of Mother Nature to give this topoisomerase. It's got these great properties just to this one species. So he was puzzled, and he and a colleague said, I wonder what the distribution is of these essential proteins in the whole tree of life. So they went looking. Now, before we get to their answer, let's do a prediction, all right? Let's say this is true. Let's say this picture is true. And down here, the last universal common ancestor has a DNA genome. Using DNA, so it's going to need a topo 1, it's going to need a topo 2, and because they're essential, everybody else should get a copy. Essential hardware present in that ultimate ancestor for all of us, we should all get a copy of those same proteins descending through the tree of life down to us. That's the clear prediction from this picture and what we know about biochemistry. So this French guy went and looked, and every time I show this, I get a little annoyed because he says, we should expect one topo 1 and one topo 2 throughout the whole of life. Why? Because an intelligent designer would have done it that way. Okay, this is offloading on your competitor theory, your own failed prediction. An intelligent designer can do whatever he pleases, right? This is taking your failed prediction and projecting it onto your competitor theory. In fact, he says the actual pattern is much more complicated. And there it is. Right, this is a figure from their paper. There are so many different topoisomerases in life that you end up with this spaghetti bowl pattern. You can kind of make out the 3 domains. They're down, buried underneath there. But look at all these different proteins doing that essential job. In fact, there are so many that they postulate ancient viral reservoirs to give rise to them. Deus ex machina, right? There's no evidence for that other than we need somewhere for this protein to come from. So some ancient virus had it and donated it to the tree of life. Okay, this is what I would call a visual reductio ad absurdum of the single tree picture. There's no single tree here, at least in terms of these proteins. There's a hodgepodge, and you might as well get rid of that single tree. Another puzzle. How many orphans are out there? All right, what's the trend? Well, this group, in a little over 10 years ago in the UK, they said, we're going to investigate this. And they said, there seems to be no sign of leveling off. We're going to keep on discovering these orphans. So here's a figure from their paper. And notice that neither of these lines is coming down. The reason there are 2 lines here is these are the long orphans, and these are the shorter orphans with a cutoff at 150 amino acids. But neither of these lines is coming down. And you'll notice there's a big jump here. When these species were done, they had a lot of orphans, so you get a big jump in the line. Okay, so there's a backstory to this diagram being in this talk. In 2005, I gave a talk at Dartmouth College in New Hampshire, where right now, you know what's going on in New Hampshire. I don't need to go into it right now. It was well attended, and there was a microbiologist from the Dartmouth faculty sitting right in the front row. And he sees me put this figure up, and he gets really agitated. So the Q&A arrives, and he shoots his hand up. He wants to ask the first question, and he's really upset. He says, Paul, what's in the numerator position? What's the data that was used to make that figure? I said there was 122 genomes. Okay, and then he gets real self-righteous. He says, what's in the denominator position? And I said, I don't know. And he said, you don't know. Nobody does, but it's some really big number. There are millions of species of bacteria on this planet. We've only sampled a tiny portion of them. This is a classic mistake in statistics. You do not generalize over the big domain on the basis of a tiny sample. You are bound to be wrong. He said, you know why orphans look interesting? It's a sampling artifact. You just haven't looked enough. Look further, look in greater depth. These orphans are going to go away. Well, what could I say? It wasn't my paper. I was citing these guys. It was their study. That was their sample. I had to swallow my pride and say, well, you might be right, but you know what? He was wrong. And I want to get back to Dartmouth. I hope he's still there because I want to show him he wasn't just a little wrong. He was really wrong. Okay, so 4 years later at the University of Connecticut, a couple of guys looked at an interesting puzzle. They said, what is the pattern of orphans, of these unique genes in the bacteria? So they took 573 bacterial genomes. There were a lot more available at the time. And they asked them a question. In effect, sampling at random, they said, who else has this gene? So they took one species, did this over and over, pulled out a gene, and then asked the other 572 species, do you have this gene? Just the way that if we went to the Fargo Public Library here, and I told you, take a book off the shelf at random and find the rarest word you can in that book. So you pull a book down and you come back to me and you say, anti-disestablishmentarianism. Wow, that is pretty rare. I don't hear that all the time. I wonder what other books in this library have that word. So we sample and we find that only 1 or 2 have it. The, of, and, basic high-frequency English words are going to be in all of them. Some of those words are only going to be in 1 or 2 books. So what they discovered was amazing. They said, there are so many unique genes that you will never see them all. Meaning every time you sequence a new bacterial species, you will see genes you haven't seen before. And they represented their data this way. Now, I'm not going to spend a lot of time on this because I can see that it's already 8 o'clock. But what I want to show you is this big green ball here. These are the unique genes. The smallest ball are the ones that everybody has. And as we sequence more bacterial genomes, this little blue ball gets smaller and smaller and smaller. And this green ball grows faster and faster and faster. And in fact, what they said is the domain of genes within the bacteria is of infinite size. Now, that's hyperbole, right? There are a lot of bacteria on this planet, but it is not an infinite number. What this really should be is it's so big we can't see the outer boundary, but it's not actually infinite. Anyway, here's how it works. Here are the genes that everybody has. It's a little bucket, every bacterial cell, that is, right? And we've seen them before, so they're not that interesting. Then they have a kind of middle category where they say, well, these are genes and proteins that are for some specialized niche where the bacteria might find itself living. This bucket's a little bit bigger because it's not going to be true for every bacterial cell. But here are the genes that are unique, right? And that bucket is enormous. And because of the way you count, this bucket grows a lot faster than the other two, meaning we will never exhaust these discoveries. This green ball is going to get enormously big compared to the other two. So, sorry, I hit the wrong, that's what I want. The number of orphan genes vastly exceeds the number of known gene families because of the way that we count them when they're discovered, and there's no reason to expect this to change anytime soon. It's really the most remarkable finding of biology, I think, in the past century. So, the genome of Luca. So if this is a little evolutionary tree, Art Chadwick is a friend of mine at an Adventist school in Texas. Some of you may know him. And I'm not saying, by the way, I'm more evolutionarily advanced than Art Chadwick, but I am younger, all right? So I put myself up there. But here's a very, very simple cartoon evolutionary tree showing the relatedness of all of life going back to Luca. The problem is, if there are a lot of orphans out here, the genome of Luca gets biologically unrealistically large very quickly. And this has come to be called the Genome of Eden problem because there's no way that a single cell deep in the history of life on this planet could have given rise to all the genetic diversity that we see. But there's a related problem. The number should go down, all right? So Mr. Human Genome himself, Frank Country. Raised the U.S. government to be the first group to sequence the human genome. He said, I don't buy it. He said, there must have been thousands of common ancestors. Again, motivated by genetic evidence. But let's look at these guys. All right, which I love is walking their outdoors. I don't like to see them going across my kitchen floor. Nor does my wife. Right? In 2010 and 2011, 6 ant species had their genome sequenced. Here are 3 of them. Okay? And you picked these 6 because they had either biological interest to us or they were in agricultural tests or something. But we've essentially picked 6 species at random from the tree, the evolutionary tree of ants. Now, when they were initially sequenced, they had very high fractions of orphans in their genomes. These numbers came down because we have to compare these to each other. When you do that, the fractions come down. So this is revised down to 11, roughly 10%. This one, 5.6.
Is that better? Okay, here are the other 3. And again, we picked them because they're interesting to us. If you've ever seen a leaf cutter, go to Costa Rica or Mexico, you see a leaf cutter in action, they're amazing. A population of leaf cutters can strip a bush overnight, and they march back to their nest with their little piece of leaf. They were done, and again, the fractions were real high, then they came down because we have to compare these species to each other. But we can take even these revised numbers, and I want to point out before I do, we're just drawing at random from this evolutionary tree. We've picked these 6 because we think they're interesting or they bother us for some reason. But there's no reason to think we just happened to pick the 6 that had really high fractions of orphan genes. But let's just take an average. We'll take an average from these 6, and it comes out to about 10% per species. And again, there's nothing unusual about these 6 species, as far as we can tell. We'll run the numbers. Just within the Myrmecinae, which is a large group of, thought to be related by evolution, a large group of ant species, they have about 17,000 genes per genome. At a 10% fraction, you come up with nearly 10 million unique genes just within that group. What happens when you extrapolate that to all 14,000 ant species worldwide? So let's say this is the size of the genome of the common ancestor of those ants, according to evolution. All right? It's a box 130 inches on a side. All right? It's a square 130 inches on a side. Let's put it down there. And this is the orphan gene number. A box 258 feet on a side. And these are obviously not to scale. Meaning, if you run the numbers for ants, the genes that are unique vastly outnumber the ones that are shared. And what happens to this picture is these nodes of common ancestry fracture. They bust apart because you cannot pack that many genes into a common ancestor for these groups. There'll be way too much genetic diversity here to fit into this kind of tree picture. And this tree will break apart at its base, which is happening already. But they're everywhere. The water flea, Daphnia, had its genome sequenced. Well over a third of its genes are found just in Daphnia and nowhere else. This little tick, and when I get to heaven, I'm going to have a little conversation with God on a quiet afternoon when nothing else is going on, and he's in a good mood. I'm going to say, what did you have in mind with ticks? Because I really can't stand them. And we'll also, if he's amenable, we'll discuss lima beans. When I got old enough to say no to my parents, who, for reasons best known to them, loved lima beans, I said, I will never eat another lima bean, and I never have. That was 40 plus years ago. Okay. But these little ticks, an enormous fraction, over 70% of its genes, at least right now, are found only there. Salamanders regenerate their limbs. Wouldn't that be a great thing to be able to do, right? You could have duals
It might be painful, but you know, after the duel, the guy chops off your arm, you just go home and you wait a while and you grow another arm, all right? That would be a wonderful thing to be able to perform as a biological function. Some salamanders can do that. That function appears to be mediated by orphan genes, meaning that unique feature of this species is provided to it, at least in part, by a gene that only it has, and on and on and on. But here's the kicker, and I'll end with this. You've been very patient. Evolution has a speed limit or a distance limit. Let's go back all the way to the start of the talk and walking to Honolulu. If Luca actually existed, and there's an evolutionary process here that connects, or processes that connect the rest of life to Luca, there's an outer radius that extends from that point that we can define. You have to be alive, right? So you can't have an evolutionary change that kills you. You've got to be able to leave offspring, and you've got to be able to perform the normal functions of life. We can define that radius and say that's all the farther you can go in 3.5 billion years. If you can map that sphere and say, if Luca existed, this is the protein and genetic diversity that we should expect to see on this planet. That's not the space available to an intelligent designer. An intelligent designer can go wherever he, she, it pleases because it's not bound by any kind of material process. So if you see a cool protein somewhere out here that you want to give to a salamander so it can regenerate its limbs, you don't have to march there from Luca. You can go right there. This is a place at which the predictions of Darwinian common descent and intelligent design differ dramatically. And what will happen in the next decade is the data will settle the debate. Because I think, and I think this is happening already, we are discovering that this sphere that extends from Luca is far too small to accommodate what we're discovering, which is actually out here. And the genetic diversity of this life, which is actually also the protein diversity, because these genes are coding for structures that will do things in cells. We're discovering, I believe, that this space is well populated and it's way beyond what evolution can do. So this is a place at which the expectation from evolutionary theory and the expectation from intelligent design diverge dramatically, and we're in a great position that we can let the evidence settle the fight. Well, the dispute. All right, you guys have been really patient. I went over my time, but I do have some time for questions if you want to throw them up here. Thank you. Yeah. Right. Is that, should I take that as your question? Okay, I'm going to repeat it for the sake of everybody. Early in my talk, I said that a key aspect of science is that it's falsifiable, which means, quite simply, that if you say something about the world, you have to be able also to say how you would know if you were wrong. In other words, I make a prediction, and you can go out and check, right? And it's not a prediction like it's going to rain tomorrow or it's not going to rain tomorrow, right? That can be true no matter what. I've got to make a risky prediction that could turn out not to be true when we check it. All right, I agree with you on that. And the gentleman's question is, it looks like I'm arguing that intelligent design is true because evolutionary theory is false. Okay, I would not want to leave you with that impression for a number of reasons. First of all, the people that I was quoting, Venter, Kunin, Didier Raoult from France, none of those guys accepts intelligent design. In fact, Venter and Kunin are strongly opposed to it. I don't know about Raoult. My guess is he is too. So the number of possible positions at play are not exhausted. That number is not exhausted by Darwinian evolution on the one hand and intelligent design on the other. So from the patterns I'm describing, you don't go directly to intelligent design. To fully make the case for intelligent design, you would need to bring more to bear. And I had an hour and I ran over that, right? So I'm sorry if I left you with that impression, but that's not my view. And my own view is intelligent design rests on a variety of evidential bases, one of which is this kind of pattern. But just saying evolution, a particular theory of evolution fails, you can't go directly from there to the truth of intelligent design. That would be a logical fallacy. So I agree with you about that, and I'm sorry if I left you with that impression. Doing science is more than tearing down the other guy's theory, right? So let's say I hire a contractor and I buy a piece of property with a little old bungalow on it, and I want him to build me a McMansion, which happens in my suburb in Chicago all the time, right? So I come back 6 weeks later, he's sitting in his truck drinking coffee, and there's nothing there. The ground is perfectly smooth. There's no house. There's no McMansion. So I go up to him. I say, "Where's my house?" He says, "Well, I got rid of the one that was there." And I say, "You don't understand, right? Getting rid of that old bungalow, that's the first step. But if you're going to do science, you've got to do something of your own on that ground. You're going to build a house. That's a different story." So I'm very glad you brought that up. It's an important point. Intelligent design, to be a scientific theory, needs to make risky predictions of its own. And because I had a lot of ground to cover tonight, I wasn't able to do that. I only barely touched on it with these last 2 slides, and I apologize for that. Did that help? Okay. I saw a hand right here. Right. 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 Yeah, let me comment on that. It's another excellent question. I don't think the universe of possible causes carves up neatly into natural versus supernatural. I mean, we can make that distinction, but think about what we're doing right now. Okay, we're having a conversation. If we recorded that and represented it as a string of ones and zeros, which is in fact how it is represented electronically in my laptop or back there or on the web, let's give that to an alien intelligence. They don't know about us. They just get this string of ones and zeros, and we say, decipher it, decode it. They subject it to various analyses, and they say, this appears to record an exchange between 2 intelligent beings. Then they would be right. Now we say, explain it in terms of physics, strictly natural processes. Explain it bottom up in terms of what physics can do. And they say, we can't do that. No physics, no set of physical laws will let us explain how we could have this conversation. For that, we need Paul Nelson, and what's your name? Sonia. We need 2 agents. Now, what we're doing is not miraculous. It's not supernatural. But in an important sense, it's not physical either, in the sense that we could bypass you and me and still explain our conversation. To explain it, we need an irreducible agent. You, another one, me. And even if I had your brain fully wired up, I could not predict what you're going to say next. That's the mystery of free will. I can't know. There's something about our agency that escapes physics but still is perfectly real. So in the universe of possible causes, it isn't just natural, it isn't just supernatural or some dividing line between them. There's this notion of intelligence that's very powerful, it's very real, and it leaves effects that can only be explained by intelligence. Everything around us in this room, the chairs, the lights, the building itself, these devices, to explain them, you need intelligence. It's not spooky or miraculous, but neither can you explain it using only physics bottom up. Physical laws and regularities are very strong and very dumb. Okay? I could drop this microphone until all of you are bored and go home. We all know what's going to happen. It's going to fall towards my hand at a certain mathematically well-defined rate. Dropping a microphone into my hand or using physical law will never give me a microphone. So intelligent design says we can use this notion of intelligence to explain, because you know what? We do it all the time. Every day, everybody in this room, to get through your day, you have to be able to say that pattern had a cause that had a mind like mine. That one physics can handle, right? Even something as simple as going to a parking lot, go to a movie, you come out, there's a note tucked in the window. It says, I'm sorry, I bumped you. Here's my phone number. Here's my insurance information. The very last thing you're going to do in a situation like that is sit down and start running through all the possible natural causes that can leave a note in your car, underneath your wiper. That would be deeply irrational because we know that that's caused by intelligence. So I'm so glad you asked that, because a lot of people have this view that it's got to either be an undirected physical process, no mind, or it's spooky, supernatural. No, there's this third category right in the middle that is part of the basic toolkit of human rationality. We all use it. The question is, can we use it for something like orphan genes? That's what makes intelligent design so controversial. Does that help? Okay. Other questions? Did I? Yeah. No. You mind if she has her follow-up? Okay.
Besides supernatural, could there be?
Well, in the full context of everything I believe about the universe, I believe that the designer of life was the God of Abraham, Isaac, and Jacob. I'm a Christian, okay? But at Discovery Institute, where I'm a fellow, I interact with other fellows who are Jewish, agnostic. I've interacted with Hindu design advocates. In May of last year, I was in Ankara, Turkey, lecturing to about 600 Islamic students, most of them persuaded of intelligent design, and their designer is Allah, right? So if we're going to have a science of intelligent design, we have to have a form in which people with different religious convictions can work together on the place where they agree. So for that reason, I want to say intelligent design only gets you to the generic notion of an intelligence. It does not give you the identity of the designer, even though as a Christian, my candidate is the God of the Bible. I'm just being honest about that. But the this notion of supernatural should not be an obstacle to your thinking. Think about it this way. If we could bring back Aristotle or Plato and sit them here, right, and show them my laser pointer, all right, can put a bright green dot on a screen, why shouldn't they regard that as supernatural? They have no categories to understand how, holding something in my hand, I can put a really bright green dot on a distant point, or show them a TV remote, or tell them that we can fly 5 or 6 miles above the surface of the earth, eating steak, listening to fine music, and dozing in a very comfortable chair, and go to another continent in the space of 5 hours. For them, that would be ludicrous magic, yet we do it all the time. So we don't actually know what the upper bound on our ability to do things is. It's actually kind of scary. Homo sapiens were very clever little bipeds. And that line between natural and understandable and supernatural kind of moves all the time. It's very scary when you think about it. So to me, the design of life required a transcendent mind. But it's still a mind that I can recognize as being a mind in some sense like my own. So I don't know if you do any art or music or poetry or create anything, but when you write a poem or write a piece of music, you bring into the universe something that did not exist before, that can only be explained by you, that's perfectly real, but no physics will ever explain. And I find when I interact with atheists and agnostics about this, and I try to take away their own creativity from them, if I'm interacting with somebody who writes music, for instance, they get really upset really quickly. They know just how hard it was to get that piece of music to sound beautiful. And they want to claim that creativity for themselves, and they should. It didn't come from the universe, it came from them. So I don't think you should let the adjective supernatural sort of stand in the way of pursuing what might be true about the universe and surprising and turn out to be perfectly good science.
The design possibilities you are mentioning if you go back and back, was it one possible design and it changed to many?
You're asking about the history of life, right?
In a way, yes.
Yeah.
Because if you allow me, then I will ask you other questions.
Sure. At Discovery Institute and among intelligent design advocates, there are a variety of viewpoints. So some of my colleagues think that the first cell was, in a way that we don't really understand right now, front loaded with all the information that you would need to unfold over the history of life to give rise to everything. That's not my view, but it is held by some designers.
So what is your own view?
My
Go
own
back to as far as you can. Say with the Big Bang.
Well, I don't think I couldn't go back to the Big Bang because, as I said, answering a question a few moments ago, physical laws and regularities described by mathematical relationships seem to be information poor. They don't give you what you would need to build a cell.
But as human beings, we have come to know we see patterns in everything. As a result, we learn what they are, and then we learn the scientific aspects of those. So what is these design possibilities that we cannot accept that scientifically we can say this is this, this is this, this is that? It has to be, the way you're telling me is supernatural. The way you're showing that circle is design possibility is outside, and if I say this can be thought of as the Earth and the rest as the space, so then that definition of evolution is wrong because the evolution is from outside. If we accept, I don't accept the Big Bang, but even if we believe Big Bang, that means evolution started from the moment of conception until now. So that means we don't have the knowledge outside Earth evolution.
This is actually not meant to be the Earth. This is a hypothetical space. In the center of that would be Luca.
So the design possibilities outside Luca is only a possibility, only a speculation.
No, not at all. Let's say that I look at a cell. I'll give you a concrete example. Okay, free copper is a toxin. You do not want copper ions sloshing around inside your cells because they're highly reactive and they'll create all kinds of mischief. So take that fact. A, copper is a poison. Fact B, copper is an essential cofactor in many enzymes in your biology. You have to have it. So how can that be true? How can you have a poison that's also essential? There must be a system that binds copper, that transports it and releases it in a controlled way. So you
exactly scientifically told me that you can actually have that poison through certain things.
So
right.
So
if we just, I'm sorry I'm cutting you off, but I have to because I've watched all your documentaries, all your books. I read everything about it, and always I thought, which today, of course, I got the chance to be here. The design possibility is like if God created us through whatever religion, I'm not atheist either. If God created all of us in that manner that the religions are saying, then that's unfair, and he, she, whatever it is a very bad designer, because not only there is design defects in us, but in our mental conditions is bad too. So then it's not just. So that's why the theory of heaven and hell comes. So I have to wait for that to be seen that I'm judged to go in there or not. So when the design comes, then these design possibilities makes it unfair. I be more intelligent than them. How can this be? So if we take evolution, at least you can say there are parts of evolution we don't know, like the Orphan genes, as well as redundant genes, as well as we don't know what they are. So that means it has given me and everybody else that capability to do what and become what we want. But that design put me in a category that I am the slave of my genes.
Okay, sir, you've raised a lot of issues. I need to take a few of them. You have raised issues that are perfectly real and important about, for instance, the imperfections of human biology. Now, I disagree with your interpretation that that we are manifestly imperfect, but that raises theological questions about the nature of the designer that I have not introduced tonight, and they go well beyond the subject matter of the talk this evening. They're good and important questions, but to address them, we would really need a whole nother seminar. May I go back to the point you raised earlier, though, because I wasn't able to finish my point about copper. In biology, you can know things that indicate that you need some other system to make the whole thing work, right? So I said, it's poison, you need to have it. From those, you can triangulate your way. There must be a system that binds and releases copper in a controlled way. You should be able to do that for design and go out into the space and predict proteins that would have to be there on the basis of what we know about cells. It would be scientific because it would be a falsifiable prediction. You wouldn't go off into mysticism. You wouldn't go off into intuition. So
What we design,
we do it.
that is predetermined by something or someone. Look, I've done a lot of design in electronics and microelectronics. I really panels for screen. One of these panels, which were intelligent panels, could see me as their intelligent designer. None of these panels could see that. And I designed other things as well. As a designer, I always wanted to see, because especially people with religion, they sit there for hours and pray, and I say, but you are not in the same domain as that supernatural being that you consider in different shapes. The same way, how could that panel or table or chair could see me as its intelligent designer? The same way, how can we see that intelligent designer of all things? How can we describe it? Because if you do that, then you are the designer, and then who created the designer?
These are all excellent questions. They are philosophical and theological in character, and I wish we had a week to debate them. I'm sorry.
If you tell me this, that how to bring justice and fairness in design if it is predetermined?
I would say that our very sense of justice and fairness does not have an evolutionary base. Evolution doesn't give a damn. Pardon the expression. No. Evolution. All Darwinian evolution cares about is making babies. You make babies, you're good to go. And And you can find examples in nature, in mammalian populations, a male will take over the pride of another male, defeat it in direct conflict, and kill all the cubs. There's a good Darwinian explanation for that. We would never tolerate it. We Homo sapiens. I'm sorry?
I said what you are saying about all these things are, yes, over time of this has changed as well. The fashion of it is changed. The first woman president may be there. The first Jewish president may be there. So we are changing. We are transforming.
But I think we have.
It's not designed.
No, I would disagree with you. I'm sorry. I think what we find distinctive about human beings, our sense of morality, our sense of right and wrong, we do not find in any other species on this planet. It makes us who we are. All right.
Yeah.
They're dumb and they have no sense.
Oh, I have tremendous respect for animals, but well, you and I are just hitting a tennis ball back and forth at this point, and we should probably
and thank you so much for this. Is great.
Your questions are first rate.
Legitimate.
I just can't entertain all of them tonight.
Absolutely no. And just really briefly, if you'd like to come back the next couple of nights, we'll actually touch on a lot of these issues, right? A lot of these questions. The doctrine of sin, for example, is something that we might touch on, right? Which is fine. Which is fine. Right. Okay.
I'm not agnostic. I'm not atheist either. I just let that window.
Right. Great. Well, if you want to come back and try it later this week, please do.
God is not so far. But that was the time that unfortunately we didn't know about genes and DNA. As time has passed, we have changed. We have got more ideas and more.
Right.
I see a question over here.
Right. Thank you. Yeah. If you want to come back, thank you so much. Yeah. And I think this will be our last one because it's past 8:30. But
so one of the takeaways that I got from your talk tonight is that there are scientists now who, based on the data, on the evidence, are starting to reject common descent. What are some of the theories? What are they looking at now if they reject common descent and maybe Luca? Where are they going now?
One of the possibilities that Carl Woese was developing before he died was a picture of the history of life where there was never a single cell, never that Luca entity, but a large, unimaginably large population of cells that never shared common ancestry. In other words, they arose independently of each other, that were exchanging genetic information back and forth. And when the three domains emerged, they did so independently of each other. So the eukaryotic domain, the bacterial domain, the archaeal domain, those three major branches arose independently out of this prebiotic milieu. Woese actually had a paper with the title The Universal Ancestor. He said, there never was that cell. So that was his picture. In the case of the French microbiologist, his view is very similar, but even more radical. In the case of Craig Venter, he says there must have been thousands of these independent starting points. One of the reasons you can't go directly from the orphan data to intelligent design is if you raise the probability of the origin of life. In other words, if you think of it as a bead on a wire with 0 never happening, 1 bound to happen, move that bead away from 0 over towards 1, you could have multiple independent origins of life on this planet that are not spatiotemporally connected. In other words, life is bound to happen given the right starting materials on that view. And just the way that quartz will crystallize here in North Dakota and give you a hexagonal prism, and you'll get the same geometry in Sweden and the same geometry in South America, given the right starting point. the picture that many of these scientists have is sort of the independent crystallization of life in multiple locations, where some things will be the same, but there'll be a lot of diversity. In Darwin's own time, this was a very popular view of evolution. Darwin was very much the exception in the 19th century for saying that there was just this single tree. Most evolutionary thinkers in the 19th century, and even into the 20th century, had a view of multiple independent starting points. So to get to intelligent design, you need to bring more to bear evidentially than I've been able to do this evening. I really wanted to tell you the news about this discovery because in the next decade, it's going to be increasingly significant to our understanding of the history of life and all kinds of other things, and people need to know about it. So if you put my name in a Google search box and orphan genes, you will see I'm widely criticized as being this kind of shameless cheerleader for orphans. And yeah, I am, because it's the most interesting thing biology has learned in my lifetime. And I just can't help but tell people about it. Does that help a little bit? The idea is, if it's natural and it could happen more than once.
Give it up for Dr. Nelson.
Thank you for coming on a cold Tuesday night.
Yeah, thank you. Thank you so much for coming out. We really appreciate your time and your presentation. Just as a reminder, we'll be back tomorrow night. And actually, we'll have a little video that kind of segues us into tomorrow night. If you want to throw that up there really quickly. There are programs at the end that Pastor Brooke is handing out, just so you know what the week will look like. But we want to have you guys back. Please come back tomorrow. We're going to talk about the purpose and the meaning of life. What is it? We have a video to show you about that here in just a second. Thank you so much. We'll see you guys tomorrow, hopefully. Invite friends.